Good afternoon, everyone. We'll get started in a minute or two. Okay, we're going to go ahead and get started. Good afternoon, everyone, and welcome to the NINDS Biomarker Program Technical Assistance webinar. We will start off with a presentation of slides followed by questions and answers. Please submit your questions in the chat section featured below, and they will be answered at the end of the presentation. We do ask that you please remain muted at all times and please keep your video off. This presentation will be recorded and placed on our YouTube page in the upcoming days, and I will be sending that link along with the presentation presentation slides to everyone that has registered. I will now turn it over to our program director, Dr. Carol Taylor-Birds.
Thank you, Rakonda, and thanks everyone for joining. I understand my audio has been a little bit an issue, so I'm going to turn my video off. So as Rakonda said, just to introduce you to myself and the team. So I'm Carol Taylor-Birds. I'm the program director in the Division of Translational Research at NINDS, and I hold the program grants for the Translational Biomarker funding opportunities. Dr. Ram Ardachandran is the program director for the Translational HEAL Initiative Biomarker Program and also holds the paying grants for our translational NINDS portfolio. In addition to Rakonda, who you just met, we also have Dr. Adisa Silu, who's a health program specialist with the team. And today we are delighted to have Dr. Natalia Strunikova, who is our guest speaker, who's going to be talking about the new review framework that NIH has launched, and she'll be talking about that and what it means for the review branch towards the end of the presentation today.
Great, thank you guys.
Thanks for joining, Natalia. So just briefly, we're going to go over the NINDS Translational Biomarker Program goals, some important terminology to be aware of, the funding opportunities themselves, and then the NIH data sharing policy, just a reminder about that. And then we'll turn it over to Dr. Strunikova. And then just one last update, we had a webinar last week on the new digital health technology derived biomarker, PAR, and so there's a slide in there just for people to be aware of, and you can check that information out on the website. So the Translational Biomarker Program at NINDS is geared to facilitate the validation of biomarkers and biomarker signatures as fit-for-purpose tools for use in clinical trials to accelerate therapeutic development and aid in decision-making for patient care. So what does that actually mean? A lot of the research we fund, as you're probably aware, is really in terms of understanding the biology of the disease and condition, and maybe for understanding fundamental knowledge about the biological or pathological processes in different diseases and conditions. So that's in contrast to the translational funding opportunities. And we have three sets of these: the discovery and development, analytical validation, and clinical validation. And these funding opportunities are really geared towards helping establish the evidence that's needed in order to use biomarkers for decision-making, either in clinical practice or in therapeutic development. And that's what we're going to be talking about more here today. And so when you're thinking in terms of, is this a good fit for your research? One of the questions is, what are the deliverables? If it's really more to understand the biology and the disease, then you're really more what we call the R01 space. Whereas if you're really thinking about how do you get this into use in a clinical trial, then you would want to come talk to us. So just make sure everybody's on the same page here. A biomarker is defined as a defined characteristic that can be measured as an indicator of a normal biological process, pathogenic process, or response to an exposure or intervention, which may include therapeutic interventions. And this is from the NIH FDA Biomarker Working Group BEST resource. So just to find that, within the funding opportunity, we also talk about composite biomarkers or biomarker signatures. Right now, the FDA and NIH are working on kind of clarifying the terminology around these, but this is essentially a combination of multiple variables or biomarkers that are combined. And so you'll see these terms used a little bit interchangeably. We'll also be talking about a detection method, and this is essentially the method used to measure the biomarker, and that might be imaging or assay, an algorithm, so on. And that's just a link to the FDA workshop that was last held back in 2022, when they started talking about the terminology for the composite biomarkers. So two more critical terms that you want to be aware of is the concept or concept of interest, and this is the aspect of the individual's clinical, biological, physical, or functional state or experience that the assessment is intended to indicate or reflect. So the biological process of the biomarker would be a concept of interest. And then the other one we really want to highlight is the context of use. And this is a statement that fully and clearly describes the way the biomarker would be used, and it has important considerations when it's going through regulatory context as well. And we really emphasize the context of use statement in these funding opportunities, which are required in all of the translational NOFOs. And we're going to talk about them a little bit more. I just want to highlight this publication that came out in 2017 from the FDA and NIH Biomarker Consortium, and this highlights how biomarkers are used in the FDA therapeutic development framework and walks through some of the evidence that's needed when regulators are thinking about how to interpret biomarkers within clinical trials. And just highlighting that for the purposes of these funding opportunities, we're talking about all therapeutic development, not just drug development. So this is a little bit more information on the context of use. I'm really thinking about how will the biomarker be used, what's the category, we're going to talk about those in a moment, and then when the biomarker would be used and how would it be used, co-decisional, enhancement, etc. And of course, what is the intended population? So really important here for these funding opportunities within our NINDS Translational Biomarker Program, the context of use statement is going to help reviewers understand why the research is important. So what is the unmet need? How would these be used? Why are these tools needed? And it also helps reviewers evaluate whether the research strategy is appropriate. Are you designing your study to generate the data you need in order to use those biomarkers and figure out how to interpret them for the context of use proposed? So I'm not going to read all of these. Links to this are in the slides, which will be shared, and these are also available on the BEST glossary. But different types of biomarkers include diagnostic, monitoring, pharmacodynamic response, predictive, prognostic, safety, and susceptibility risk. And this just briefly highlights sort of the general ways that these different categories of biomarkers are used in therapeutic development. And there's always some exceptions to the rule, but essentially thinking in terms of what phase of clinical trials they could be used. So two more terms before we get into the funding opportunities. Analytical validation is a process to establish the performance characteristics of the test, tool, or instrument are acceptable in terms of its sensitivity, specificity, accuracy, precision, and so on. And clinical validation is a process to establish that the test, tool, or instrument acceptably identifies, measures, or predicts the concept of interest. Again, that term that we used before. So now let's get into the funding opportunities themselves. So We have now reissued all of the translational biomarker funding opportunities. I want to point out that the PAR 25050 and PAR 25055, these are the U01s for analytical and clinical validation. These were reviewed or reissued now with the new simplified review framework that Natalia will be talking about at the end of today's presentation. The SBIR U44s, this doesn't apply to that, to the new NIH policy around the review change, but all of the other funding opportunities do. And then PAR 25024, which is our development R61, R33, our earliest stage funding opportunity, was also fully reissued this summer.
Carol, my apologies. I don't see the slides. Is it everybody's seeing them? I don't see them. Are
No,
you sharing them?
I am. Has that been true?
Yeah, I can see them.
I don't see them.
Anyone else not see them?
I see a comment, they see the slides as well.
Anyway, I don't see the slides.
Okay. Wakanda, do you want to chat with Natalia and see if we can figure out what's going on the
Sure.
file? Thanks.
Mhm.
All right. So also, just as I mentioned before, the new digital health technology-derived biomarkers and outcomes UT3 UH3 was also just published this fall, and we had a webinar on that one last week. And that will also be available online, the recording of that, and the NINDS Biomarker website as well. So just briefly to go over the funding opportunities scope. So for this first development PAR, the really critical point here I showed before, sort of the overlap in the R01 and translational programs in terms of the biological process and fundamental knowledge. But the really critical part of the discovery and development, in addition to having a context of use statement and thinking about the translational aspect of the biomarkers, is this initial proof of concept clinical validation study. So the R33 phase needs to have a clinical proof of concept for the context of use you propose. Then the analytical validation, U01 or U44s, is really about saying you've already established that there's a good reason to have these biomarkers, and now you want to optimize and establish the performance characteristics of how you measure the biomarker. So essentially, can I trust the results of the measurement? And what are the limitations of those measurements? And then finally, the clinical validation is really a large prospective, usually study that you're going to evaluate the biomarker utility for the context of use proposed, and whether they're meaningful and interpretable for making decisions at the individual participant or patient level. So thinking again about how the biomarker is used, if you're going to be recruiting patients based on the results of the biomarker, what's that threshold for determining whether they're included to the trial or whether they're included to a particular arm or not. So thinking about the action from the readout of the biomarker as well as just the association. And finally, biomarker qualification is not required for these funding opportunities, although we certainly encourage groups to think about pursuing that with the FDA. If the biomarkers context of use is for therapeutic development, that could be broadly used with multiple therapies. But again, this is not necessary for all context of use or all projects. So what about preliminary data and premise? So what are what we call the entry criteria for one of these funding opportunities? For the discovery and development PAR, or R61, R33, you really need preliminary data and supporting literature, that there's a strong biological rationale for why you think this biomarker or biomarkers is appropriate for the biological process or pathological process that you're interested in. And then Or if you're doing more of an unbiased approach, are you going to identify it? What are your selection criteria? And so on. For the analytical validation, you're expected to have already done an initial proof of concept, which demonstrates that you can, there's evidence that the biomarker has sufficient potential value to use it as proposed, and that you now want to do additional optimization of the detection method. So really strong preliminary data are expected here. You need to have already developed the detection method. So you must have at least a prototype, and you could do some additional optimization. This is not to develop new assays or detection methods. And then you want to make sure that you're also including, you might be including the standard operating procedures, evaluating pre-analytical variables, and so on. And then in clinical validation for preliminary data and premise, in addition, you essentially have to have already established all of what you would be expected to develop in the development and analytical validation PARs. So detection method has been analytically validated, you've got the proof of concept clinical study, and now you're going on to do the additional final clinical validation. I think I had a duplicate slide in there, sorry about that. All right, so research scope. So what are you actually proposing to conduct and do in these funding opportunities? So the biomarker development PAR, again, this needs to include a plan for doing the preliminary clinical validation proof of concept. You can include detection method development, but that can't be the focus of the grant. You need to also make sure you include that clinical validation study. It can be multi-site, but it's not required. And the analytical validation one, sorry about this, my slides are out of order, can include the optimization of the detection method. You can include multi-site validation study design, and that's actually expected, so you need to say why it's not needed if you don't propose to do multi-site validation. And you can include the analytical validation of the detection methods, really looking at rigorous evaluation of the performance characteristics, establish your standard operating procedures, and so on. And then finally, the research scope for the clinical validation is to establish the statistical thresholds needed to interpret and make decisions about the biomarker within the specified context of use. So again, what would be the threshold, or what would the criteria be if you get the results of measuring the biomarker? How would you use them? What's the appropriate definitions and criteria for making a decision, whether you're going to diagnose someone or stratify them into a particular trial, or determine if they are more responsive to a particular therapeutic, so on. Now, just some basic characteristics and requirements for these funding opportunities, and this is to compare across all three. In the discovery and development PAR, the R61, R33, the first phase can be 1 to 3 years. The second phase can also be 1 to 3 years, but the maximum project period can only be 5 years. There are milestones between the R61 and R33 phase. The budget's not limited, but it needs to reflect the project period or the goals. Intellectual property is not required, but if you're proposing to commercialize the detection method, then you would want to include an IP plan. And then with all of these, the research, resource, and data sharing is expected, so you need to have a plan for that. With the analytical validation, note that this is only a 4-year project. So for the U44 phase for the SBIRs, you can see the years there as well. And again, these have, with cooperative agreements, they have annual milestones each year of the grant that need to be included and proposed in the funding in your application. Same with intellectual property for all three of these. If you're going to commercialize, then you should have an IP plan. If you're planning on doing open source sharing, then that's not required. And again, data sharing is required for all of these. And then finally, the clinical validation, essentially the same, except the maximum period for the research is 5 years. So non-responsive studies, you really want to make sure that you go through the criteria that are in the PARs for each of these. These are the ones from PAR 25024, the R61, R33. And if you have any questions about this, please reach out and ask. We do have to administratively withdraw anything that are non-responsive. And so we don't want people to come in and waste their time with the application if they're not going to be in scope. This is really important as well. You know, we said that the budgets are not limited, but they need to reflect the actual needs of the project. If you need to go over 500,000 direct costs in any 1 year, you need prior approval to come in, and that's a NIH and NINDS process for doing that. So you need to contact us at least 6 weeks before the applications are due. And then we go through an internal review process and we'll need some information, and that information is available on the NINDS website as well, and can share those links also. Finally, the timeline and proposed milestones. So all of these NOFOs require go-no-go milestones. For the R61 and R33, it's between the 2 phases. For the cooperative agreements, it's annual. You want to make sure that these are quantitative, these are clear on what the deliverables will be. And for each milestone, you want to provide a brief justification of what the criteria for success are and why you are proposing those justifications. And an important note about this as well is that these get evaluated as part of the overall approach by reviewers. And so you want to think about, are these appropriate? How does this fit with the rigor and expectations of your research strategy overall, as well as thinking about the timeline feasibility. So a little bit more on the milestones. These can include things that are quantitative performance. They can also include things that are sort of standard progress. In either case, you want to be really clear on what the success criteria are and why. You also want to make sure you're including the timeline, and including something like a Gantt chart can be helpful for that. A couple of, so these are not new for the UO1s anymore, but for all of the PARs, R61 and R33, as well as the UO1s and U44s, you need to include a team management plan. This is included in the other attachments rather than in the research strategy. And this helps outline how the overall team will work. We also have the plan for enhancing diverse perspectives. This is also required, and you really want to think about what the different types of diversity are on your team and how you're utilizing those within the research overall to achieve the deliverables of the project and how they're being incorporated. If you're proposing to use research from existing clinical trial, just make sure that you're including the trial protocols and consent forms. And then again, intellectual property, you can include if you're planning on commercializing. And of course, letters of support are expected but not required. I'm not going to read through this in the interest of time. This is just more information from the funding opportunities and the team management plan and the plan for enhancing diverse perspectives. And then we're going to just touch 1 more minute on the data sharing plan, and then we'll turn it over to Natalia. And Adisa, do you want to talk about the data sharing plan?
Absolutely. Thank you, Carol. So another important aspect of the application is the data management and sharing plan. NIH has a commitment to making any results of any funded research available. And so any applicants that are planning to generate scientific data will need to submit a data management and sharing plan. These plans are not going to be part of the scored peer review criteria, but will be reviewed and approved by the program staff. So NIH as well does expect, the investigators and institutions to plan and budget for the managing and the sharing of the plan, which is also included in the plan. And after submission of the plan that is approved, comply with the approved DMS plan. Slide. Next slide, please. Thank you. So the data and managing sharing website has a lot of information when it comes to the policy. Anything regarding any data and sharing management, as well as sample data managing plans could be utilized to write your plans. Next slide. One important fact as well is having access to the facts site, which will allow you to answer any questions necessary. Next slide.
All right, and this is just the PAR we mentioned before, the new one. This will be in the backup slides when we share the slides later. All right, thanks, Adisa. And now we are going to switch over to Natalia.
Yes, hi guys. My name is Natalia Strunikova, and as you probably understand, I am coming from the review side of things. I am managing NSD translational review study section, and many of the biomarker applications are as a standing study section. So we're meeting 3 times a year, and we are reviewing a lot of biomarkers applications. Specific to the variety of neurotrauma conditions, hypoxia, stroke, cardiac arrest, pain, sleep, tumor development, neuropathy, and cognition. So if you have any questions about the review side of things, you can always contact me. And this is a kind of summary about me, and this is what I do, but I did it for many years. Next slide, please. Thank you. I'm going to speak about what is coming in January, and going to highlight some thinking process behind the simplified framework for the NHP review process. It's still work in progress, although it's the note is published already. But of course, it's an interaction between the applicants, reviewers, review and program sites. So we're still facilitating how we do it best, so everybody understands the goals and the processes at the best possible way. So the overall goals of the changes and what is behind the changes is to enable peer reviewers to kind of better focus on answering or evaluating the key questions necessary to assess scientific and technical merit of the application. So basically, to highlight that, we're asking them to overview the application from the point of view, should this proposal or this research be done, or can this proposed research be done? And these are like very or kind of 1,000-feet questions, but this is, we're trying to bring the reviewers as close as possible to the evaluation of the science and the importance and the impact of that particular application, rather than focusing on kind of minutiae of programmatic things that are now shifted to the program stuff in many instances. We're also trying to mitigate the effect of the reputational bias, and I'm going to go over it in some of the slides which are coming up, but basically we are trying to make sure people are kind of assessed, or PIs who are applying assessed based on their access and to the materials and the environment and the ability to lead the program, rather than kind of how well they're known in the field and if they are kind of known better than other PIs. So that would be one of the goals of the changes. And we're also trying to reduce reviewers burden. And this is what I mentioned previously. We're trying to shift up regroup some of the things from 5 criterion areas into 3 kind of groups of review criteria. And let's see if I can highlight how it's going to be done. Next slide, please. So it's just to reiterate what I said already, but kind of from a different point of view. So 5 review criteria that we have right now in this section 5 of every opportunity announcement, and are going to be reorganized into 3 factors, and some additional review criteria as inclusion of certain human subjects will be moved into factor 2, which is will be known as rigor and feasibility, and this factor will be scored as well as factor 1, and again, we're going to try to reduce the reputational bias, because we're going to basically package the investigation and environment assessment into factor 3, which is going to be factored into overall impact score, but not going to be scored separately. It's going to be more or less a binary assessment, meaning is it kind of sufficient or not sufficient, and if there are any gaps. And if there are any gaps, you will have the reviewers will have an opportunity to kind of comment on the gaps and what could be needed and what is going to be required to fulfill those gaps for the APIs or for the environment to perform at the best possible or expected level for the completion of the project. So the reduce review burden, we'll already mentioned that we're going to shift a lot of stuff from additional review consideration from the reviewers to the NIH stuff. Sorry, guys. And some of the things are going to be moved to the factor 2. It will be scored together with factor 2. Next slide, please. This is basically the summary slide, which I'm not expecting you to you can look through it, but probably you will assess it or look through this multiple times during the writing process of the application, especially when you write the research strategy of the proposal, because the research strategy section of the proposal needs to be kind of the way you lay out things needs to be aligned with section 5, which contains the review criteria. And they need to kind of have a conversation between each other. So certain things that, for example, program is expecting from you needs to be present in the research strategy, and then it needs to be present in a form that it kind of answers the questions that reviewers are going to be utilizing to assessing the data presented in application. So just to make sure we are highlighting all the factors, we're going to have 3 factors now as a part of simplified research criteria. Factor 1, it's importance of the research. Basically, it's a significance and innovation that are now combined into one factor, because they, in many cases, to a certain degree, they overlap. And again, that particular factor should help the answer the question, should the proposed research be done? And significance and innovation are grouped together to actually simplify or kind of streamline answering to that question for the reviewers. Innovation is much broader defined in the for that particular factor, and so there is a room for reviewers to evaluate how innovation is influencing the importance of the research, how impactful the research is with many of the innovative factors, and what type of interplay between significant and innovation. So this particular factor is going to be scored from 1 to 9, and going to be that particular score is going to be feeding into overall impact score. Then we have factor 2, which is we call now rigor and feasibility, and also that particular factor includes approach, what used to be approach in 5 criteria. It's also going to be scored from 1 to 9, the same thing. It's going to feed into overall impact score, and it's included as well, including as well inclusion of women, minorities, and for all the projects, a study timeline for the clinical trials. Rigor is highlighted very thoroughly in this factor, and a lot of language is going to be that is going to be used is similar to what we had previously in the approach. And I'm going to give you the slides, next slide, so you remember. You again, you can go through this. So the reviewers can focus on the quality of the work and attempt to answering the question, can the proposed research be done? So again, this is a really kind of 1,000-feet view. Really, can the proposed research be done? So we're actually focusing on the science and how impactful the science, and do we have enough to actually perform that particular scientific project? If you can go to the next slide, that would be the factor 2. Yeah, so it's a lot of bullet points, and when I'm not going to read you the bullet points because it will time to processor, but I kind of giving you the idea behind it, so you understand the kind of strategy and the logic behind it. Feasibility also a part of it, because we by asking all these questions with asking reviewers to evaluate the risk. How is this possible to do the projects? Do we have everything lined up? And milestones are also part of it. Okay, so the milestones, although they're not scored separately, they are going to kind of fit into overall assessment of factor 2, which is going to be scored 1 to 9 based on the discussion. And again, it's going to fit into overall impact score. Now we're moving to the factor 3. The next slide, please. Yeah, that's the feasibility. So which called now expertise and resources. And we're kind of putting a lot of spotlight on the expertise and resources, including evaluation of investigators and environment, as it's what used to be criterion 1 and 5. But here we're asking the question and trying to determine whether the PI has the necessary skills and resources to perform the study in the context of the proposal. And it's a part of overall impact score, and reviewers needs to save the whole package of investigator and environment and technologies, and interplay between all of them is considered to be appropriate or not, but there is no separate scoring. So basically it's a binary assessment, and as I said, there is going to be an opportunity to provide a feedback if certain things are missing, and you guys are also going to be able to see it in a summary statements. The next slide, please. So as we mentioned, additional review criteria are going to be reworked somewhat, and as we're anticipating that the reviewers' burden will be lightened by that. So we are moving some of the things into the factor 2, and some of the things are going to be moved under review, consider like they're going to be reviewed by the program side instead of the review panels. So on the right, you can see revised additional review criteria that are usually and typically influencing the overall score, but they don't have their own kind of scoring 1 to 9 matrix, but the comments or the issues or deficiencies as strengths in these 4 bullet points can influence the score, overall score during the discussion. The next slide, please. And but in terms of additional review considerations, we're having a slightly different situation where this particular review set of questions don't bear any significance on a score. It's also, but it's also a very important part of the discussion, and it's used to be a very important part of the discussion. But now most of them are going to be moved to program staff, and the program staff going to evaluate them separately. We are going to retain authentication of key biological for chemical resources part and budget and period of support for the reviewers to look at. But again, these two things are not going to influence the score. Thank you. Next slide. This is the summary slides for you guys. So to look at it, what we had before January 25th and what we're going to have after. So this is very kind of simplified comparison of two strategies, kind of the typical 5 criterion scores situation and the 3 factor grouping of the review criteria. So if you have, like, keep it in mind and utilize this table when you're writing the application, just to make sure whatever you're putting in the application is consistent with the review criteria that that are present in your particular proposal in the section 5, because this is what reviewers are going to use. And I think this is it for me.
That's
Right?
it. Thanks so
Yeah.
much, Natalia.
Take care.
All right, so finally, just want to remind folks that we have lots of resources on data sharing. These are links to some of the repositories that you might want to check out. And this is the information which we shared in the slides deck as well, if you want to join any of the listservs. We're going to go ahead and stop sharing now, and we are open to questions. Thanks, everyone. I think Adisa's going to moderate the chat.
There we go. Oh, sorry about that. I thought you could hear me. So we do have one question that states, do you have a list of already qualified biomarkers, if any, that are related to brain disorders?
Yeah, thank you for the question. So there is not any qualified biomarkers as yet. And one of the slides that we have in the backup is linked to the FDA's Biomarker Qualification Program. They have put in a nice website now for drug development tools. I'm going to throw that in the chat right now, where you can look and see where some of the projects are. There's a 3-stage process, a letter of intent, submitting the 2nd stage qualification plan, and then the final data qualification package. So this is a pretty intensive effort. We do have a project that's in this process, but right now we don't have ones that are qualified. Now, there's lots of resources from the FDA to learn more about that process, but just making the point that for groups coming in developing a therapy, they may use their biomarkers for their therapeutic development and have those approved as part of the therapeutic product approval process. And that's distinct from sort of the open source, open available biomarker qualification program, where you're trying to develop something that's going to be used for multiple therapeutic indications. So I hope that's helpful, and the link is in the chat there.
Thank you. The next question is, what is the total funding allocated to biomarker program in 2025?
Right, so these are not RFAs, these are PARs, and I know we have lots of terminology, but an RFA has set aside funds for the funding opportunities. A PAR does not. So these have special review criteria and requirements, but these are coming out of the overall NINDS or NIA funds available. So the same pool of money as our R01 or other non-RFA mechanisms. So we set a pay line and funding recommendations each year for the R01 space. Traditionally, historically, the NINDS Biomarker Program more or less tries to match that funding plan. Obviously, if funding is limited, there can be some contingencies there, but essentially we match the funding rate of the R01 space.
Thank you. The next question is, do you consider feasibility from FDA regulatory perspective as part of the review process?
So
Yeah, go ahead, Carol. Yeah.
yeah, so it depends on the context of use, right? So it's information that's provided to reviewers as well as to all of the applicants, but it's a consideration when the context of use is for a therapeutic. And then Natalia, did you want to add something there?
Yeah, I was just trying to say this is a standing panel which reviewing only translational applications from early on to the very late stage. So we do have experts who are knowledgeable about FDA regulatory process, and they actually trained to do it for many years. So they're rotating over the course of the years, but we do have a sizable number of people that are coming from even the companies. So they're not even from universities. Yep, thank you.
Yeah, thanks, Natalia. Yes, we've got great review panels.
Absolutely. The other question is, what are the restrictions on applications that include foreign partners' site?
Yes, important question. So applications, we won't, we cannot accept applications from foreign organizations as the contact organization, but you'll see this in the requirements for the funding opportunities. Foreign components are allowed. So you can have a collaborator from a non-US institution that needs to be justified and explained in the application, but those are allowed.
All right, that's all the questions I have currently in the chat.
All right, and we'll just give it a couple of minutes. I know we're pretty close to the end of time, but if people are still thinking and have questions, feel free to throw them in the chat. Or if you want to raise your hand and ask a question instead, we can do that now as well. Again, all these slides will be shared and the recording will be shared after the webinar in a few days. And if you want to discuss a project and think about whether it's a good fit or not, you're always welcome to reach out. We ask for a draft aims just to kind of help guide the conversation. But please do let us know. And I just want to remind everyone that our development PAR R61, R33 NIA is also signed on to. All right, thanks everyone. Really appreciate you joining, and thanks to all my fellow panelists. Take care. Have a good evening.